Hello all, welcome to Life Science Library. In this video, we are going to see about another topic from Plant Physiology Unit 6 Iicsirnet Life Science portion. It is phototropin. It will be very useful for GATE aspirants as well. You are watching this on Life Science Library. First up, we are going to see what is phototropin. Phototropin is nothing but a flavoprotein. It acts as a blue light photoreceptor. It is predominantly present in nucleus. It is a membrane associated protein and it has no membrane spanning domain. The N-terminal portion of this phototropin N-terminal portion of this phototropin has FMN and the carboxy terminal has serine threonine kinase. This N-terminal is called as photosensory domain. It has two love domain, LOV1, LOV2. LOV indicates light oxygen voltage, that is called as LOV domain. These are the basic details about phototropin. Now we will draw N-terminal domain. It has photosensory region of LOV1, that is light oxygen voltage, LOV1, LOV2. Then we have serine threonine kinase. It is a C-terminal domain. We have attachments like FMN, in the N-terminal end of both LOV1 and LOV2. This occurs in the photosensory domain. This is a photosensory domain. This is the basic structure of phototropin. It is a serine threonine kinase domain. It is a carboxyl domain. N-terminal domain is a photosensory domain. Next, we will see what will happen in the presence of light and in the absence of blue light. First, we will see what will happen in the absence of light. When there is no light. Absence of light. In case of absence of light, we have N-terminal domain. We have LOV1, LOV2, two domain. Serine threonine kinase domain in the carboxy terminal and attachments of FMN, attachment of FMN with LOV domain. When there is no light, when it is completely dark, when there is no light, there is no conformational changes and phosphorylation does not take place. No phosphorylation. There is no conformational changes. In dark. This is the situation of dark. We will see what will happen in case of presence of light. Consider in the presence of light. What will happen in the presence of light? Blue light. We have N-terminal domain, LOV1, LOV2, light oxygen voltage, serine threonine kinase domain at carboxyl end when the light is irradiated at the photosensory region. So light is irradiated in the photosensory region. They form a covalent bond between FMN and love domain. Covalent bond is formed between FMN and love domain. And the serine threonine kinase is underwent autophosphorylation. This occurs at the carboxy terminal. This is the mechanism of action once if this phototropin is exposed to light. There are two types of photo. First type is photo 1, next type is photo 2. These two photo are associated with accumulation responses and avoidance responses. Photo 2 will exhibit avoidance response, photo 1 will exhibit accumulation response. Let's see what is avoidance response and accumulation response. Before going into that, why phototropin has to accumulate something and why phototropin has to avoid something? Because phototropin has a major role in regulation of photoperiodism and chloroplast movements. Why chloroplast has to move? To prevent photo damage. What is photo damage? Take for example, if we have a leaf here, we have so many cells, chloroplast cells. Consider they are irradiated with high intensity of light. So, when it is exposed to high intensity of light, photo damage will occur. Photo damage will occur. If the photo damage will occur in a region where there are in the direct exposure to light, where they are directly exposing to light. So, if this is a leaf, and few cells are present here, and light ray come like this, the much affected portion is the cells that are directly beneath the source of light. They receive very high amount of radiation, so they undergo photo damage. The role of this phototropin is to prevent photo damage, and they also involve in chloroplast movement. So this is the reason why chloroplast move. Chloroplast move to protect from photo damage. This is the reason why chloroplast move, and this is the process by which photo damage occurs. So now we will see what is accumulation responses and what is avoidance responses. Let us see. Let us consider two cells, two plant cells, cell 1 and cell 2. Let us consider that we have so many cells here and there. Okay, and consider that this cell is irradiated by light source. This is low light. Very low amount of radiation comes from it. So what will happen? As a result of this, cells started accumulating on both the sides. Since the light source is very low, cells started accumulating. This kind of response is called as accumulation response. This kind of response is called as accumulation response. So now we will see what is avoidance response. Consider this as a cell. And consider we have so many chloroplasts here and there inside the cell. Consider a light source. Of very high intense light source. High light. We are exposing the cell to high light response. So what will happen? As a result of this high light intensity radiation, these cells will tend to protect itself by avoiding to getting itself exposed to high light radiation. So they will tend to avoid that light by moving to the sides. This is called as avoidance response. This condition is called as avoidance response. So, Photo will undergo avoidance response. We have another component called chloroplast unusual positioning 1. So, what is chloroplast unusual positioning 1? Chloroplast unusual positioning is a protein. They help in attachment of photo to the cell membrane. Consider they help in anchorage. Consider this as a cell. Consider this as photo 1. Consider this as photo 2. We have CHUP, that is chloroplast unusual positioning 1 protein. Chloroplast unusual positioning 1 protein is present. And then consider this as a chloroplast on which photo 2 is present. This is CHUP1, chloroplast unusual positioning 1. It is attached with N terminal. It is attached with N terminal side. Basically, it is associated with anchorage. It is a protein. Consider this as photo 1. This as photo 2. Photo 1 and photo 2. Chloroplast unusual positioning 1. Help in anchorage. Now we are going to see about various mutation cases. Fort 1. What are the duty of Fort 1? Fort 1 is associated with cotyledon and hypocotyl growth. Fort 2 can withstand high light intensity. In case of double mutant, they provide a specific blue light response. Fort 1 mutant lacks hypocotyl photoperiodism in response to low light intensity. but retains phototropic response at high light intensity. If Photo2 mutate, they exhibit accumulation response and there will be no avoidance response. If Photo1 mutate, there will be avoidance response. There will be low accumulation response. This response is very low if Photo1 mutate. What will happen if both Photo1 and Photo2 mutate? There will be no response. What will happen in case of low light intensity? Movement to the upper and lower side, that is accumulation response, where maximum light absorption takes place. What will happen in high light intensity? High light intensity movement parallel to the incident light takes place. It is a protectionist measure. Overall, this is the basic understanding about phot and phototropin. Phot1 and Phot2 are the two different types of phototropin. Let us sum up this video on phototropin. Phototropin is a flavoprotein. It is a blue light receptor. Previous year question. Then what are the components will be present in N-terminal site and what are the components will be present in the C-terminal site? In the N-terminal site we have love domain, love 1, love 2. C-terminal we have serine threonine kinase domain. They have FMN in both the cases. They also have the LOV is lowest light oxygen voltage. What will happen in case of blue light? In case of blue light, N-terminal side low 1 and low 2. Serine threonine kinase in carboxy terminal. In case of blue light, there will be covalent attachment with FMN. Covalent attachment with FMN. And serine threonine kinase will undergo autophosphorylation. This is the mechanism. Phototropin are of two types, Phot1 and Phot2. Phototropin has a role in preventing photooxidative damage. This is a previous year question. Two types of responses. Phot2 avoidance response, previous year question. Phot2 will avoid. Phot1 will accumulate. This is a previous year question. CHUP1 is a chloroplast unusual positioning. This is also an important pointer. Mutation cases are very important. There are so many questions asked in the previous years. Phot1 associated with hypocotyl growth. Phot2 is associated with high light intensity. What will happen if Phot2 mutate? They will undergo accumulation response. Phot1 mutate. Avoidance response. Both mutate. No response. In case of low light intensity, they manage to be in the upper and lower side chloroplast cells. High light intensity, Phot2, they move in the corners parallel to the incident light. Hope so this video is very clear on phototropin and various responses like avoidance responses and accumulation responses and about all the domains and components of phototropin. Hope this video is very helpful for everyone. Thank you for watching this video. Thank you.